Hello, welcome to King's Bioproduction Academy. Let's today understand about mutation. We're getting to learn about what? Mutation. What's mutation first in the definition? Mutation is the spontaneous change in the genetic makeup of the organism. So we are saying mutation is what? Mutation is the spontaneous change in the genetic makeup of an organism. We're speaking about what? Spontaneous. Spontaneous means something that can occur very fast. And it's about what? Genetic. It's about genes, genetic makeup of an organism, of which this mutation will have a lot of problems. You understand we have two types of mutation, gene mutation and chromosomal mutation. And we have the types like deletion. When you speak of deletion, it's a certain section of a nucleotide base is being cut from a polynucleotide line. You have the adenine, cytosine, guanine, and thymine. Those alignment of those nitrogenous base. In case one is cut, maybe it will cut that gene which was very important for synthesis of a certain organ. And what will happen? That now organism will be incomplete because a section will be missing. Sometimes it can add some of those nucleotide base. If they are advantage trait, it means they will have more addition of the advantageous characteristics of the organisms. So can you let's about discuss some disorders which arise from gene mutation. So we are discussing about what? Disorders. Disorders arising from gene, not from gene but from mutation itself. Let's just be general. Which are those ones? We have those which will be sex linked and we have those which are not sex linked. Like color blindness, one, color blindness. Let's discuss which are linked to sex chromosomes. X chromosome linked to what? These are what? Sex chromosomes. They are linked to sex chromosomes. Hope you are getting this one. We are saying they are linked to sex chromosome. We have the gala blindness. Number two, we have hemophilia. Those are the ones which are linked to the sex chromosome. Color blindness, you know, it's about failure to distinguish colors. That is the green, red color. Hemophilia is the bleeder's disease. This where blood fails to clot even under minimal what? Cut. Then from that now point, we have this other one which are not sex linked, but which are those ones? Albinism. And we have sickle cell anemia. 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 On this sickle cell anemia, what's about the sickle cell anemia? Sickle cell. Even the other one we have what? We have this cystic fibrosis. Cystic fibrosis. This anemia I've written wrongly. It's supposed to be anemia. Yeah. This way. So let's understand my main concern. Let's discuss about the sickle cell anemia today. Sickle cell anemia. Let me just remove these ones. I'm saying let's understand about what? One, sickle cell anemia. What is this sickle cell anemia? This is where an individual have the recessive alleles for this condition. They have recessive alleles for the condition. That is when a person will speak about being sickle cell anemia. The red blood cells all will be sickle cell shaped. They will all be what? Sickle cell shaped. So we are speaking about when a person will speak about having a sickle cell anemia, this person has recessive alleles. They have recessive what? Alleles. And which are these recessive alleles? They are the recessive alleles, which will, when we speak about the recessive alleles, when it comes to the sickle cell anemia, when you do your genetic crossing, you'll be using HBS, HBS. This is what I'm speaking about, recessive alleles. Those S and S. The normal hemoglobin person will be having HbA, HbA. Then we have that's homozygous. Then we have now the heterozygous, that is the sickle cell trait, will be HbS, HbA, HbS, sorry. So these are recessive alleles. I hope you have understood the recessive alleles. I'll say that is HbS, HbS. What is the meaning of that HbS, HbS? This means this person's red blood cells have lost their biconcave shape and now they are what? Sickle cell shape. So we are saying red blood cells are sickle cell shape, or you speak about crescent shape, being a crescent shape. And what's the disadvantage of a person being with a sickle cell anemia? The disadvantage for this person and can only survive. This person can only what? Can only survive under what? Close medical attention. Close medical attention. What's the main reason? We are saying the red blood cells are sickle cell shaped. And being all sickle cell shaped, it means they cannot squeeze through the narrow lumen of the blood capillaries. One adaptations of the red blood cells, which are normal, are biconcave shape. And one of the adaptability feature of that will be the biconcave, so that they can squeeze through the narrow lumen of the blood capillaries. So this one cannot squeeze through the narrow lumen of the blood capillaries. And it means if they cannot squeeze, the transportation of oxygen will be very difficult. So this person under very low oxygen concentration, they cannot survive. So we are saying we'll have a problem. Have problem on oxygen transportation. And in case this person goes to a place where we have low oxygen concentration, in case of what? Low oxygen concentration, they can do what? Easily suffocate. They can easily what? Suffocate. They can easily suffocate and even die. They can easily suffocate and do what? And die. That is the sickle cell anemia. It's bad when having this condition. It's a condition and a disorder. When you speak about a disorder, we are speaking, it's not a disease, we are speaking it's a disorder. Get the difference. We have a difference between a disease and a disorder. So we are speaking about, in case of this low oxygen concentration, this person might suffocate and definitely do what? Die. That is it. So let's do the second one now. This one is the sickle cell anemia. Let's do the sickle cell trait person. We have number 2. Then we'll summarize on. So we are saying we have what? Sickle cell trait. Which are the alleles of this person? Which are the alleles of this person. Let me just rub all these on so that I can have enough space. I'm saying what? Sickle cell trait. Which are the alleles of this person? It will be HbA, that is a normal hemoglobin, HbS, that is with the sickle cell. So it means this person, they are heterozygous, have what? Have heterozygous alleles. These persons have heterozygous alleles. So it means they have what? 50% normal red blood cells and 50% sickle red blood cells. That is it. Hope you understand this one now. From this point, what you should understand is this person in the tropic regions where malaria is endemic, they get an advantage of survival. So we are saying have advantage in malaria endemic zones. They have what? Advantage in malaria endemic, or sites which are more prevalent to malaria breakthrough. So what will be the reason as to why they can survive in those areas? The main reason, we're speaking about what? Have the numbers, have normal red blood cells. So it means those have will have an adaptation of supplying enough oxygen to the blood tissues of this person. Then the half of the red blood cells, we say they are crescent shape. And because of this one, they are not pliable, hence they cannot squeeze through the narrow lumen of the blood capillaries. And because they cannot squeeze through the narrow lumen of the blood capillaries, and again the effect in those red blood cells, within those what? Red blood cells, oxygen concentration will be low. And because oxygen concentration is low because of the packaging, low packaging of the red blood cells in the hemoglobin, what will happen? The Plasmodium species, that is the Plasmodium malariae. We have the species Malariae, Ovale, Vivax, but we're speaking about Plasmodium malariae. What will happen at this point? They cannot be able to, the red blood cells themselves, they crystallize under that low oxygen concentration. And because of this one, the Plasmodium species cannot survive in that low oxygen concentration zone. And because of that, they cannot complete their life cycle. And that is why this person now will suffer from, this malaria. So we are speaking about what? In the 50% normal red blood cells will transport oxygen using its hemoglobin to which point? It will transport the oxygen using hemoglobin to the respiring tissues. To respiring tissues. That is a plus. Then now these other ones, so we'll have an advantage of not suffocating and dying like the other person. Then this one, we are saying the 50% sickle cell red blood cells will have low oxygen concentration. You're getting that. And because of having the low oxygen concentration, what will be the effect? The effect now will be those red blood cells. The red blood cells will crystallize. Thus, the red blood cells crystallize. And which are about these red blood cells which are crystallizing? Those crescent-shaped red blood cells. They are the one which crystallize. And because they crystallize, that is what? The sickle-shaped cells. Hope you understand that. Now when you have understood that they can crystallize, so what will be the effect? Thus, the Plasmodium species cannot do what? Cannot survive in low oxygen concentration. And because it cannot survive in the low oxygen concentration, what will be the effect? It cannot complete its life cycle. Thus, cannot complete its life cycle. And because it cannot complete its life cycle, it means what will happen? This person will survive from not being affected. Hence, the person attacked cannot be affected by the malaria. Hope you're getting the point now. We are saying now this person cannot suffer from the malaria because the life cycle was not completed. For everything, the life cycle should be complete for it to be infectious. But because of it cannot complete its life cycle because of the low oxygen concentration and those red blood cells crystallizing, that is where now by this person will not be affected and this person will have an advantage. Again, this person now might have some disadvantage when it comes to low oxygen concentration. The main reason, they have less number of red blood cells. They are 50%, but the normal person at least will have an advantage in case we have low oxygen concentration in those areas. So let's do some brief genetic course of this sickle cell anemia. Sickle cell anemia. So we are saying we do a brief calculation, not a genetic drawing of sickle cell. Maybe you are told cross or do genetic cross. You do a genetic cross between a normal person with normal red blood cells with the other having sickle cell anemia. So the first step that you should do is your parental phenotype. And our parental phenotype, let's say a person, not just a person, male, a male, between a male person, between male person with normal red blood cells, then with the other, that is now the female, having what? So our symbol of the male and our symbol of the female. So this one is our normal red blood cells. Then our female will be sickle cell anemia. We're speaking about anemia. So then you go to the second one, which is the parental genotype. I've said the normal is HbA, HbA. So that A is what? You're saying HB. This A represents normal red blood cells. HbA. So they are homozygous. Then you indicate your genetic cross. After indicating your genetic cross, what follows? After indicating your genetic cross, you now have this one, which is HbS because of the sickle cell, then HbS. So at the end, you'll have now the gametes here. Which are these gametes? You just bring down this one. You do a complete circle. HbA. Then this one is HbS. Then HbS. Let's try today. We use a Punnett square, the Punnett square. So the Punnett square, you just indicate this one, a square. Does not come to be that complete square. You divide to 3 first, the second slot, and the third one to 3. Then again you divide again this one 3, 1, 2, and the third. Then this one we divide in this manner, not this way. Then you indicate just the signs of the male and the female. Those are your gametes. From that point, these are the female. So it's H female, BS, HBS. For the male was HB, A, HB, A. So it will be HbA, HbS, HbA, no, sorry, HbS. This one is a HbS, HbA. We don't start with HbS, we start from the normal, then the other one, HbS, HbA, HB. So it's this HbA, HbS. There's still HbA, HbS. HbA, HbS. You can check this your fusion. And after your fusion, you get your what? F1 phenotype. When you ask it now about these, after doing that genetic cross, their offsprings. These are your offsprings. Let's say you have F1 offspring. Which are these F1 offspring? You can check all are HbA, HbS. All that HbA and HbS, they are not normal hemoglobin. They are not sickle cell anemia. So they have the sickle cell trait. So you speak about what? F1 phenotype will be all have sickle cell trait. You get your mark. Then F1 genotype, all HbA BAHBS. You get your mark? So that is the end of what we can discuss today about this sickle cell anemia. Thank you very much. We continue to learn. So before that, sorry, let me just indicate about those cells. You can have a drawing of the normal and the sickle cell. So when you speak about the normal red blood cell, it's always in this manner. That's our normal red blood cell. This other one we are speaking about, it's a crescent shape. So this one is what? Sickle cell shaped red blood cell. This one is a normal red blood cell. Thank you very much.